Hello everyone, this is another video of the chapter photosynthesis, and today we are going to discuss about the factors affecting photosynthesis. So here first we'll be going through the external factors that affect the process. So light intensity, carbon dioxide concentration, temperature, water content. So as we come to the topic that is light intensity, so when the leaves are being exposed to the light, photosynthesis occurs. But if the intensity of light increases, photosynthesis rate increases for a certain level, and after that again it comes to a constant level. Same happens with carbon dioxide concentration. In this, what we can see that photosynthesis will further increase, but again it is stabilized at a point. So the two factors, light intensity and carbon dioxide concentration, that affects photosynthesis simultaneously. Next topic is temperature. So as we know that any life process which takes place in living organism, that depends upon an optimum temperature, means a range of temperature. If the temperature decreases, means the enzymes won't be activated. But if the temperature increases or exceeds optimum temperature, then the enzymes get destroyed and the process stops at that point. Okay, so means that photosynthesis process that takes place between a range of temperature. Then comes water content. Water content, if there is scarcity of water, then the photosynthesis won't occur. But if the absorption of water is more from the soil, then the the rate of photosynthesis occurs at a higher speed. Then comes to internal factors. In case of internal factors, we can say that number of chlorophyll or protoplasm and structure of leaf that affects photosynthesis. How come chlorophyll affect photosynthesis? As we can say that if there is any deficiency diseases present in the leaf, that can affect the number of chlorophyll, or the loss of chlorophyll may occur, because of which the photosynthesis process is hindered. Protoplasm, if it is dehydrated, means there is loss of water, then protoplasm affects the process of photosynthesis. And the next is the structure of leaf. Structure of leaf, that depends upon the thickness of cuticle, distribution of stomata, and the size of the leaf, and that influence the process of photosynthesis. Next comes the experiments on photosynthesis. So let us see what all experiments we can demonstrate. Try to demonstrate photosynthesis. So first of all, the experiments which will be showing that that there is a requirement of de-starch leaf. Now let us first understand what is the meaning of de-starch. De means loss of starch from the leaf. Okay, so leaf, they loses the starch, and this can be done by the experiment that what we can do, we can leave the plant in dark for about 2 to 3 days, okay, or 24 to 48 hours. Now during this period, what happens, the starch which is present inside the leaf, that will be consumed by the plant, and as there is no light present in that room, so what happens, that enables the leaf is not able to perform photosynthesis. So the starch won't be prepared, and the starch which are present in the leaf will be utilized by it. So that means we are destarching the leaf, means removing the starch from the leaves. Now starting with the experiment 1, that is to test a leaf for starch, that can also be known as iodine test. So what we can do, we can take a green leaf from a plant that has been exposed to the sunlight for few hours. Then we'll take a half filled beaker with water. Then we'll heat the water until it boils. Keep the water at boiling point. Use a forceps to place the leaf in the boiling point for 2 minutes. Turn off the Bunsen burner and then place the boiled leaf in the boiling tube containing 90% ethanol. Now first of all, why we are boiling the water and why we are introducing the leaf inside the boiling water, that is to kill the cells. Okay, now when we are placing it in the alcohol, ethanol, or we can call it as methylated spirit, to remove chlorophyll from the structure, from the leaf, to make it hard and brittle. Okay, after that we'll place the boiling tube in hot water and boil it for 10 minutes until the leaf decolorizes. Means if the chlorophyll will be removed, then it will become pale white in color. Okay, now gently remove the leaf and wash with a fine trickle of cold tap water to remove all the ethanol which is present on the surface, and then we'll spread it on a white tile. And then to test for starch, we'll use the iodine or potassium iodide solution and we'll pour it on the leaf drop by drop. So what will view, the result will be, as you can see, this is the setup, and the result will be wherever there is a presence of starch will turn to blue black in color. Okay, so the starch, if at all it is present, that will turn to blue black in color. Means the plant has produced starch, or it has produced glucose that has been turned into starch, and that gives you blue black coloration. Okay, so hence we can conclude that the plants, they produce starch in the process of photosynthesis. Next experiment shows that to show the chlorophyll is necessary for photosynthesis. In this, what we can do, we can take a plant with variegated leaves. Okay, now what is the meaning of variegated leaves? That means there will be starch present in some part. Okay, means we can also say that different colors will be present in the leaf. Okay, so having green and some non-green areas. Now what we can do, we can destarch the leaf by keeping the plant in a dark room, as we do for destarching. Then we can place it under the sun so that the plant will start producing starch. Then after few hours, pluck one leaf, we'll pluck the leaf, and then we will test for starch, as we did in the previous experiment. Now in this thing, what we can see, that this is the result which we got. In this, what we saw, that the leaf where the greenish parts were present, that turned to blue black in color if we tested for starch. Means if we poured iodine to the leaf, so the places where green parts were there, those turned to blue black in color. Okay, and the white portion turned to brown in color. So this shows that chlorophyll is necessary for photosynthesis. Now in the next experiment, we'll show that the sunlight is necessary for photosynthesis. In this experiment, what we can do, that we'll take a plant with de-starched leaf. We'll cover one of its leaf with black paper with a design is cut. Okay, so here why we are using the black paper? Black paper is used so that the light cannot penetrate in through that black paper, and that cannot reach to the surface. Okay, that cannot reach to the leaf. Okay, now place this plant in the sun. So we'll place the plant in the sun, and after a few hours, what we can see that if we'll test it for starch, the place where the leaf was exposed to sunlight that turned to blue black color, means the place which was turned to blue black color, that contains starch. And because of the exposed because the surface was exposed to sunlight, they were able to produce starch. But the place where it was covered with black color, black paper, that were not able to get the sunlight, so the place were not able to produce starch. Okay, so that will turn to brown in color. So this shows that the sunlight is necessary for photosynthesis. So in our next video, we'll be seeing the other experiments.